Academy, Adilabad, preparing for NEET. So today I am going to talk about the topic process of recombinant DNA technology. So when we make a recombinant DNA, we will have to go through 6 different processes, namely isolation of the genetic material, fragmentation of DNA, isolation of a desired DNA fragment, ligation of the DNA fragment into the vector, transferring the recombinant DNA to the host, culturing the host cells on a large scale, and then extraction of the desired product. So first we are going through the first process, which is isolation of the genetic material. So as we know, every organism on earth has its genetic material as nucleic acid. So most of the organisms have DNA as the genetic material, which is short for deoxyribonucleic acid, and some organisms like viruses and etc. have RNA as their genetic material, like short for ribonucleic acid. So for this, now because we know that inside a cell nucleus is present, and to get to the nucleus we'll have to break the cell open. So for breaking the cell open, we'll have to use some enzymes because cells have cell walls. So to break these cell walls, now we know that different organisms on earth have different components in their cell walls. So we know that for plant cells, in plant cells we have cellulose. So we'll have to use cellulase enzyme for plant cells. Similarly, we'll have to use chitinase for fungus, lysozyme for bacteria. Then we have to separate the macromolecules. So macromolecules are RNA, proteins, polysaccharides and lipids. So to separate these, we'll have to use different enzymes like RNase, protease and lipase respectively. Then we'll get the separated DNA. Now after that, we'll have to remove it. So we'll have to add chilled ethanol to it. So after that, we'll get precipitated form, and that precipitate is called as spooling. Removal is called as spooling. Thank you.